I'm Nicole Meisberg, and I'm going to be doing Lab 1, which is practicing aseptic techniques for growing bacterial cultures. Um, First off, when you go into the lab, you want to make sure that you don't have any coats, your backpack is put away, food is put away, your phone is put away. They're all in the clean zone, which is away from the lab, so there's no bacteria or anything getting into your stuff. Or your stuff doesn't catch on fire, so that would be good. Next off, make sure you're wearing the right clothes, proper attire. No backless shoes like high heels, clogs, regular shoes that have backs, no shorts. Also, make sure that if you have long hair, it needs to be secured, braided, tied back so that it doesn't get in any of the lab bacteria, petri dishes, or a flame, because that would be really bad. And then also, no outer coats, that's put away too. What I have on this is my mock pretend lab coat because I don't have a lab coat. So it's just a long shirt. And also goggles. Make sure you have protection for your eyes. These are not actual goggles. Ordinarily, goggles would have protection on the sides so that your eyes are fully protected, but I don't have those either. So I'll just do with these glasses because they're better than nothing. So make sure that your hands are washed thoroughly before coming into your lab so that everything is clean. And also, when you get to your lab station, you don't know what's touched it, so you got to make sure that it's disinfected. So I need to clean my workspace. So you get your disinfectant and you spray it, and you use a paper towel and wipe up the workspace. Obviously not just the small section, but the whole table. All right, and then I get everything that I need for my lab. So we got 2 petri dishes, we got some test tubes. Ordinarily, these test tubes will be labeled, or you should label them before your lab. For test tubes, you should put a tape around it and label it, but I don't have test tube tape, so I'm just gonna pretend that you see one has more liquid than the other. The one that has more liquid will be the one that contains bacteria, and the one with less will be the one that's a fresh test tube that you're transferring the bacteria into. For the petri dishes, they should be labeled as well along the outside on the bottom with a Sharpie, not in the middle, so you can see what is growing. Also, for this lab, we're going to need a micro incinerator. So, you'll need to get that out and turn it on because it takes 10 minutes to warm up. And you'll know that it's warmed up when you look inside. This is my Seahawks micro incinerator. You'll look inside and see that there's an orange light in the opening. Also, you'll need your inoculation loop. This is what you'll be transferring all your bacteria into the cultures, whether it be a petri dish or a test tube. And you want to hold it like a pencil. So the first step that you'll want to do is you'll want to sanitize your inoculation loop. And if you look at the inoculation loop, not on this one, but there'll be a wire up to a certain point and there'll be a handle. You want to sanitize or disinfect that whole wire, and you do that by placing it into the micro incinerator all the way up so that no wire is exposed. So I'm going to do that and sanitize my loop, and you stick it in the micro incinerator for about 10 seconds. So after the 10 seconds is passed, it is very hot, And you don't want to stick it right into the cultures because the bacteria could die. So you want to let it sit for about a minute to cool down. So for my first procedure, I'm going to do a plate-to-plate transfer. And I'm going to take one of these Petri dishes, and usually the medium in it is like a agar that holds the bacteria. So I'm going to take it with my hand, and I'm going to slightly open it so it's cracked like a clamshell. Not too far open because you don't want to expose it to too much air and have it get contaminated. Then with my inoculation loop, I'm going to stick it in and just drag it. I don't know if you can see that. Just drag it very lightly across the surface, just skimming it in a straight line across the surface. When I'm done with that, I'm going to close my Petri dish, set it aside. Now I have the bacteria on my inoculation loop. I'm going to take the new plate to transfer my bacteria. I'm going to do the same thing and open it up only partially. So it's like a clamshell. And just do another little streak in there, just gently across the surface. All right. And I'm going to close that Petri dish. Set it aside. And this is the inoculated one. Maybe label it with a Sharpie. And now I need to sterilize my loop. So I'm going to stick it back in the micro incinerator for 10 seconds. All the way, and then pull it out and let it sit for a minute. And that is how you do a plate-to-plate transfer. Also, I forgot to mention, when you are done with your inoculated plate, you're going to stick it in the incubator, but upside down, so that the condensation doesn't get to it. I'm going to do a broth-to-broth transfer, which involves 2 test tubes of broth, one with bacteria and one with the fresh broth that is where you're going to be transferring that bacteria to. I said, since I don't have any tape on my test tubes, I said the one with more water is the one with bacteria that I'm going to transfer, and the one with less water is the fresh broth that I'm going to transfer the bacteria to. Okay, so the first step that you're going to do is you're going to sterilize your inoculation loop. I should have already done this when I finished my first lab procedure, but I'm just going to do it again for good measure. Put it in for 10 seconds, take it out, and let it sit for a minute. First, I'm going to grab the test tube by the glass, since it has liquid in it. And since this one has bacteria, we're going to mix it around a little bit so I can be sure that I'm going to capture the bacteria when I stick the inoculation tube in there. So I'm going to hold it with my pinky finger and flick it until you see some spiraling action, kind of like a little mini vortex spinning around in the liquid. So that's all mixed up. Then I'm going to take the lid off with my pinky and ring finger. So really stuck on there. Okay, got it. And since this is exposed to air now, With the test tubes, we need to sterilize the top lid because bacteria could, or anything in the air could get into this sample. So we need to sterilize the top by sticking it by the micro incinerator for about 2 to 3 seconds. All right. And once it's sterilized, then you are good to grab the bacteria sample in the test tube. So you stick your inoculation tube in there, grab some of the sample, pull it out, and before putting back on the lid, you need to sterilize the top again to prevent bacteria from getting in. So sterilizing 2 to 3 seconds by the micro incinerator. All right, and then you put the lid back on and place it back in the test tube holding area. All right, now my inoculation loop has the bacteria on it, and I'm going to take my new broth, my fresh broth. I'm going to do the same thing with this test tube. Grab it by the glass. I'm going to take off the lid with my pinky. It's really on there. Okay. And then I'm going to sanitize the lip of it, putting it by the micro incinerator for 2 to 3 seconds. All right, and then I'm going to stick my inoculation loop that has the bacteria in it into the test tube, new broth, and I'm going to kind of twist it, you see, to get all of the bacteria off of my loop so that it can get into the broth. All right, and then when I'm done with that, before closing on the cap, I need to sterilize again. Put it by the micro incinerator, 2 to 3 seconds. And then I'm going to put the cap back on. All right, stick it back. Actually, I wouldn't stick it in back where it was. I'd stick it in the incubation area of the classroom where my lab section was, the purple group. So I'd stick it in there on the rack. So let's just say I did that. All right, and then when I'm done with that, I still have bacteria on my inoculation loop, so I'm going to stick it back in the micro incinerator, making sure I got all of the wire in there for 10 seconds. And then I'm going to let it sit for a minute. And that is the broth to broth transfer. All right, I'm going to do a plate to broth transfer for this procedure. And I need a Petri dish plate that has the bacteria on it, as well as the new fresh broth test tube. So what I'm first going to do, if I haven't already done it, is I'm going to stick my inoculation loop, sterilize it in the micro incinerator, 10 seconds, take it out, wait for a minute so it's all sterilized. Then I'm going to grab my Petri dish, open it up so that it's clamshell size. I'm going to stick the inoculation loop in the Petri dish. Oh, can you see that? And just gently streak across the surface to collect the bacteria. Still holding like a pencil, not digging in. All right, I have it collected. I'm going to close the Petri dish, set it aside. And then I am going to take my new broth. I'm going to take off the cap with my pinky and ring finger, twisting it. And again, it's kind of stuck. Got it. Okay. I grabbed it by the glass too, by the way, since it's exposed to air. Gotta sterilize, put it by the micro incinerator for 2 to 3 seconds. All right, I'm ready to put in the culture. Place it in the tube, twist it to make sure I get all the bacteria off. Pull it out. And again, before sealing on the cap, I need to sterilize. Put it by the micro incinerator for 2 to 3 seconds. All right, and then twist and put the cap back on. And then I would place this in the incubator with all the rest of my lab group's test tubes. All right, but I don't have an incubator thing, so I'm just going to put it right here for now. Lastly, since I still have the bacteria on this inoculation loop, I need to sterilize. So I put it in to the micro incinerator. 10 seconds. And let it sit for a minute. And that is the plate to broth transfer. All right, so it is the end of the lab. The first thing you want to make sure you've done is you have placed all your Petri dishes in the incubator and make sure that they are upside down, so to prevent condensation, as well as your test tubes go in the incubator as well. So all of those are there. Any discarded test tubes go in a separate section, or test tubes that you are not growing bacteria on, that have waste in it, you set aside in a designated spot in the classroom for the discard area. Also, make sure not to pour any of the materials in the test tubes down the drain. Don't do that. And any Petri dishes that you have left over that you need to discard, make sure you put that in the biohazard cylinder in your classroom, wherever biohazard place you have. Make sure to put all your materials away. Make sure they are sanitized or sterilized before doing so. So if you haven't already, at the end of your lab, don't forget to put it in the micro incinerator, 10 seconds, let it sit for a minute, and then it's sterilized. Always do that. And then you want to turn your micro incinerator off. Make sure to turn that off and let it cool down all the way before putting it away. So you will notice, if you look inside, there will be an orange light that you've seen the whole time when it was on. It will slowly go back to normal and you won't see the orange anymore. And then you know it's off and it's cool and you can put it away. And lastly, before even getting up from your workstation or leaving, you want to make sure you disinfect your work area. So this is all put away. And I need to disinfect. So I use my disinfect spray and wipe it up with a paper towel. Discard of the paper towel. And my area is all clean again for the next person who wants to come in. Then if I was at school and I need to store my lab coat and my goggles, I would take them off at the end, wherever the storage area is for that. Or if there's anything I was using during the lab that needed to be sanitized, I would sanitize that as well. And then definitely after all that, wash your hands thoroughly before leaving the lab and collecting my things to go. And that is it for Lab 1, Aseptic Technique for Growing Bacterial Cultures. See you next time.